our speaker tells us he has 35 minutes for us, so without further ado, I think we should get on. It's a great pleasure to welcome Professor Jamil Inal. Jamil used to work here, then he went to Hertfordshire, and then he came back. I've returned twice to different universities, and I can tell you that going back is a thing you should do. And so I'm really pleased that he did do that. Jamil's got a very impressive CV. I'm not going to start reading it out, but I do want to point out that as well as working in those universities and having London-based degrees, he also worked at Porton Downs. So if anyone thought that that perhaps COVID was something that leaked out from under a door somewhere, I guess somebody who worked at Porton Down would know what that kind of thing was and what the risks or likelihood were. But without further ado, let's hear from Jamil about extracellular vesicle research and why it's so important or so influential in biomedical sciences and research. So the floor is all yours, Jamil. Officially, publicly, welcome back. It's great to have you here.
Oh, thank you very much.
Turn things off.
Thank you for that very kind introduction, Don. And I will eventually turn off my camera to preserve the bandwidth and so on. It's really nice to be back, and I'm going to focus the talk on the extracellular vesicles and give you a flavor of how they impact on other areas in the biosciences. So the core research has been, and this is going to be a slightly historical look through what we've done over the last 15 years, has been very much based on understanding the basics of extracellular vesicles. In other words, and that's really the sort of central bit here. It's all about the extracellular vesicle basic biology or fundamental biology. In other words, how the vesicles are made, the biogenesis, how do we actually isolate them, how do we characterize them, how do they interact with other cells, and so on. But then there are many applications of this, some of which we've already embarked on and others that I would like to in the future. So if we start at the top right here, you can see links with sports sciences. And there I would be particularly interested in looking at sort of high intensity exercised people and the kind of plasma extracellular vesicles that they would have and the kind of health benefits that they may have. And there's certain, having isolated these vesicles from exercised people, there's lots of nice in vitro experiments that we could do. Cancer biology, I'm not going to talk about that at all today, although we've had 3 very good PhD students complete their work in this area, looking at the epithelial mesenchymal transition and how this affects metastasis and so on. So that's not going to be talked about today, but it's a very important area of EV research. Extracellular vesicles in regeneration. So I have some collaborators here from a grant from a few years back, and this is based on the use of extracellular vesicles derived from stem cells and their ability to ameliorate acute organ injury, kidney in particular we were looking at. There's a lot of bioinformatics work. So there's this EVpedia, which is published in PLOS Biology. So this is looking at all the new proteins, for example, and also microRNAs and other RNA molecules that the extracellular vesicles carry. In terms of neurodegenerative disease, here I'm talking about EVs in human iPSC models of neurodegenerative disease. But in fact, we've got other collaborations with people who have a rat model of Parkinson's disease, for example, and actually a mouse model of Alzheimer's disease. And sort of a flavor of the sorts of things people working on EVs can do and why people like to have them on board their grant applications is because they are very good diagnostic markers for disease and also prognostic markers. And in fact, that's the sort of thing we're doing with the Alzheimer's disease model. This is a small project actually related to cancer, which is ongoing, and that's looking at skeletal muscle extracellular vesicles and their role in metastasis. I have another collaboration with bioengineers, and this is very interesting because it's trying to develop microfluidic systems of isolation and analysis eventually of extracellular vesicles. And lastly, working on bacterial vesicles. So bacteria also make vesicles, which is in general, vesicles, when we talk about vesicles, we mean small entities released from cells, and so they can be human cells, they can be cells, whether it be a multicellular parasitic worm or a unicellular protozoan parasite, or even a bacteria. All these entities will make vesicles. So bacteria make them too, and as with many other pathogens, they may be making them as some sort of decoy. And in this case, I'm very interested in developing bacteriophage therapy as an alternative to antibiotics. And vesicle release from bacteria may actually be influencing the effectivity of bacteriophage therapy. So the ability to modulate vesicle release is very important. And this is actually a theme running through all this work as well, and probably part of the basic work, as well as understanding the biogenesis. By so doing, you're able to influence, modulate vesicle release from cells. So I'll skip through the next slide. This is more or less summarizing the collaborations, and you can see the main topics here. So EMV biogenesis, infectious disease, drug delivery, regeneration, and tumor microenvironment, EMT, and metastasis. So a really good example of interdisciplinarity, and this is something that could perhaps be possible with the School of Computing. I have spoken to somebody working in physics and astronomy who has a way of resolving galaxy clusters, which you see here on the left. And to my mind, if you're able to do that, then you could perhaps apply that to resolving vesicle clusters, as we see on the nanocyte tracking analysis. This is a still from a film made. Normally these vesicles would be oscillating because of Brownian motion. And just as you can resolve galaxy clusters, well, why not do the same with especially exosomes, which tend to cluster quite a lot? That's a specific type of vesicle. So I was introduced to this subject in October 1999 when I visited a lab in Basel in the University Hospital, and I was shown this paper. I remember being talked through this paper. So on the top left here, you see a resting PMN neutrophil, and it's being stimulated with 1 micromolar of FMLP, and you can see these filopodia, pseudopodia being appearing. And then if you close in on the surface, you can see what they termed ectosomes, but these are extracellular vesicles, and probably because of the size, they're actually exosomes or small extracellular vesicles. And these are budding off from the plasma membrane, from the surface of the cell. Here, the recovered vesicles have been labeled with an antibody, an immunogold antibody that recognizes complement receptor 1, or CR1. And that same antibody is then used to precipitate the exosomes, and they're fixed onto dynabeads in this case. And this close-up, if you're able to close up, to close in, and actually the way there is a magnifying glass on the top left, you should be able to see the lipid bilayer. And this is the same sort of work, again with a PMN. This time it's stimulated with sublytic complement, and you can see the beginning here after 2 minutes. It's beginning to vesiculate, and then if you close in on the surface, you can see the vesicles budding off. And importantly, an hour after the stimulus, the cells return back to the resting state. Now, extracellular vesicle terminology is something that's changed a lot over the time that I've been working on it. So having that paper in October 1999, I ended up working in that lab from February 2000. And in 2005, I conducted the first experiments. Actually, it was through one of my project students, and he was isolating urinary extracellular vesicles. And the terminology was quite different in those days. So in this slide, I've tried to show you what the current terminology is. Let me just switch off my camera. So we have here the largest kind of extracellular vesicle are the apoptotic bodies. So we call them large extracellular vesicles. The microvesicles, or medium-sized EVs, as they should now be called, are 100 nanometers to 1,000 nanometers in diameter. and the exosomes, or small EVs as they should now be, as you can see, they're sort of the size of viruses, are up to 100 nanometers, 50 or 30 perhaps nanometers, up to 100 nanometers in diameter. Now here we see the biogenesis of these vesicles, both the exosomes, which are the small extracellular vesicles, and the medium-sized extracellular vesicles or microvesicles. So here at number 1, you see endocytosis of surface receptors and molecules, and this results in an endosome, which may be degraded by fusion with a lysosome, or which itself may undergo inward budding, which is what's happening here, to produce these intraluminal vesicles, as you can see here. This is now called a multivesicular body, and it can return to the surface, fuse with the plasma membrane, and release this cargo of what we now term exosomes, or small extracellular vesicles. There's also vesiculation going off, for example, from the endoplasmic reticulum, which can release molecules to the surface. But the other subject of which most of this talk is about is the microvesicles, or medium-sized EVs. These pinch off, they're shed from the plasma membrane, as we see here, during early apoptosis. So they can carry surface receptors and some nucleic acid. And when the vesicles leave the cell of origin, some will enter the bloodstream, and indeed they're found in all body fluids, for example, cerebrospinal fluid, urine, and others. So cellular pathways to medium EV release. Basically, this requires some sort of increase of intracellular calcium. So calcium influx via, for example, membrane attack complexes or P2X7 gated calcium channels. That leads to an increase of intracellular calcium, resulting in activation of cytoskeletal associated enzymes, which will cleave the actin cytoskeleton. Intracellular calcium can also originate from organelles, in particular the endoplasmic reticulum. And then I won't go through all this because of time, but it's just safe to say that a lot of vesiculation is associated with very early and recoverable, actually, apoptosis. So in this slide, we see transmission electron microscopy here and fluorescent microscopy on the sides of a cell in its resting state, and in close-up here. And then here, one that has been treated with sublytic complement. This is forming pores in the plasma membrane, not enough to cause lysis and ultimately death of the cell, enough to activate the cell to release, and you can see here these small arrows are showing some vesicles being released from the cell. There's also depolymerization of the cytoskeleton going on. So here we have stained HeLa cells with phalloidin at a fluor 660. This stain stains actin, and as you can see, during the microvesicle release here in the middle, the fluorescence is punctate compared to the non-induced cells on the left, or during induction when the cells are treated with calpectin, because that inhibits microvesiculation. Essentially, this confirms that during extracellular vesicle release, there is depolymerization of the actin cytoskeleton. Now, here we see stimulation of the cells with BZ-ATP, ATP, and calcium ionophore, and this is used to induce microvesicle release. Here, the gray bars are showing how we can inhibit this release using, for example, calpectin and EGTA. The release of these enzymes is inhibited after 45 minutes incubation with calpectin, 20 micromolar, and the cells were stimulated to produce EVs in the first place using normal human serum as a source of complement proteins. EGTA was also used as a negative control. And here the cells are stained with DAPI vector shield and annexin 5 FITC. Now, we were also able to knock down the CAP-NS1 calpain, and this inhibits microvesicle release. So basically, if we just skip straight to the slide here, we can see that the untreated cells are releasing quite a lot of microvesicles, and that using CAP-NS1 silent RNA abrogates this release, as does the calpectin, which inhibits the calpain enzyme in the first place. So these are all important tools to be able to do our biological experiments as we do them. Now here we can see extracellular vesicles released with increasing amount of stimulation. And here we can see the inhibition of this with, we know that this is due to sublytic complement because this is complement protein C9 depleted normal human serum. And you can see there's no vesicle release compared to the serum without removal of C9. In the old days, we used to put these vesicles straight onto the flow cytometer, and we didn't have, there didn't exist machines like nanocytes or nanocyte tracking analysis, which is what we see on the right. So this is a forward side scatter plot, and of course, because of the relatively poor resolution of these flow cytometers, you're missing a large portion of the population of vesicles. In fact, when we compared the same samples, we are missing in the order of several hundredfold. And so you can see here, this is what we nowadays need to use to, one of the tools to characterize the extracellular vesicles is the nanocyte tracking analysis, which this histogram gives a modal peak size, which you can see it's slightly more spread out. There's even multiple peaks for the medium-sized extracellular vesicles, but for the small extracellular vesicles, it's really coming in at about 100 nanometers. And then if you can also then calculate the y-axis is showing the concentration of the extracellular vesicles. This is showing the exposition of phosphatidylserine on the vesicle surface, and this is detected with an Exin5, fluorescently labeled. Nowadays, we have to also show that certain markers are present on the small extracellular vesicles, such as CD9, TSG101, and so on, and that they're absent on the medium-sized extracellular vesicles. In the early days, we showed that there was enrichment for actin. So these two lanes are showing vesicles derived from THP1 monocytes compared to the parental cell. So here, after stimulation, the release of microvesicles or medium-sized extracellular vesicles increased. You can see this line here, referring to the y-axis on the right. So the microvesicles increased over a 20-minute period without any further increase up to 1 hour. That's in A. During this time, and coincident with the increased numbers of released microvesicles, we were able to look at the percentage of early apoptotic cells, which you can see here as the white bars. And this almost doubled from 4.5% to 10% in Exin5 positive cells. And of late apoptotic cells, well, that increased significantly from 1 to 3% here, the black bars. That's for an Exin5 positive and 7AAD positive cells compared with the unstimulated controls. And when the stimulus was removed and the cells were assessed with 30 to 60 minutes later, that's in panel B, the percentage apoptosis levels returned to initial levels at 7% for early apoptosis and about 1% for late. This suggests that the phosphatidylserine exposition on the outer leaflet of the plasma membrane, which we detected with an Exin5, is an early feature of microvesicle release and doesn't induce cell death. And that, in fact, upon returning the cells to a non-microvesiculation condition, as we see in B, the raised PS levels are found to be only transient. This work is looking at the quartz crystal microbalance. You can see here what it comprises is a gold electrode which is oscillating. It literally weighs things. It's a microbalance, as it says, and it's oscillating. And if something is deposited on it, when you switch off the current, it stops oscillating. It will dampen down, and you can calculate using the Sawbury equation what the mass is that's deposited on it. So in this case, you've got, I think, what it looks like antibodies, and it's picking up some virus. What we did was to look at the cells that were vesiculating, and we started the cells. So here the cells are depositing on the electrode. We add the stimulus for microvesiculation, and over the course of 15 or so minutes, you see a decrease in mass of the cells, and there's an increase of rigidity of the fluid around them, and this sort of coincides with the 20 or so minutes that we saw of increasing vesiculation in this experiment. So calcium is very much a theme in all of this. So here I just put together some different agents that were increasing the intracellular calcium, which is measured with Fura-2-AM. So this is histamine, all-trans-retinoic acid, and PMA, which is a 4-ball ester. And here you can see that they are all causing an increase of release of microvesicles. And here in C, we can see again the calcium release, but this is done with a intracellular parasite called Trypanosoma cruzi. And here in A, we have the metacyclic trypomastigote, and in B, we have the epimastigote, which is the insect form of the disease of the parasite, which will not be interested in the human cells. It doesn't attach to them or interact with them or enter them. So there's no change in the intracellular calcium there. In C, we have non-induced cells. In D, we have the metacyclic form, which here caused an influx of calcium, but then treated with EGTA. And this is the epimastigote with EGTA. And in D, we can see the microvesicle release from T. cruzi, metacyclic trypomastigote, and that's reversed with EGTA. Here, another thing all of us doing at the moment is doing proteomics on these vesicles. And here, this is actually comparing what sort of proteins are found in vesicles released from that have been stimulated versus ones that have not, and that are just constitutively releasing extracellular vesicles. So here the stimulus was sublytic complement, and I think that's what we're seeing coming off here. This could potentially be the C5 beta 9 being removed in the vesicles that were stimulated with sublytic complement. And here, when you use lots of different techniques to look at these vesicles and to measure their size, here you can see quite different average diameters. So this is using transmission electron microscopy. This is using nanosite tracking analysis. This is using dynamic light scattering, and this is using flow cytometry. And here, what we see here is, again, going back to the quartz crystal microbalance, the medium-sized EV subtypes, that's the constitutively released one versus the stimulated one, were analyzed for their ability to quench oscillating momentum on the Q sense using F3. The constitutively released vesicles, the amplitude phases out significantly faster than the stimulated vesicles, indicating that the constitutively released ones were deposited on the sensor faster than the stimulated ones, and suggest that they may have a larger mass than the stimulated ones. There we go. That's the use of the quartz crystal microbalancer. So, summarizing so far, we can say that microvesicles are released upon stimulation of cells with sublytic complement, calcium ionophore, BZ-ATP, and also during early apoptosis. There are also increases of intracellular calcium, actin rearrangement. Cells secrete increasing amounts of vesicles over a period of 20 to 30 minutes after the stimulus. Of course, that's recoverable as well. And the medium-sized EVs, their release can be inhibited with calpectin or by knocking down expression of calpain. We've done quite a lot of studies looking at the biogenesis pathways and indeed finding new ones. And so there's quite a lot of other inhibitors that are available, which is quite useful going forward. The basic EV properties, they are rich in phosphatidylserine and they're enriched for actin. And most of the medium-sized EVs are between, say, 100 probably to 500 nanometers in diameter. So are we actually working with, this is something that concerned us a lot between 2007 and 2012, as to whether we were working with microvesicles medium-sized EVs, or exosomes, the small EVs. And I remember reviewing a lot of papers where there's a lot of confusion, and you often just look at the method that was used to determine what sort of vesicle population people are actually working with, because they've not named them correctly. So this is one method to ensure that you've got the small extracellular vesicles or exosomes, and that's to put them on a sucrose gradient. And here you can see in pool 1, coming out at about 1.04 to 1.05 grams per mil, we have the LAMP1 positive small extracellular vesicles, which we can see here by TEM. And we know that these are exosomes because, or small EVs, because of this artifactual cup-shaped morphology of the vesicles that we have here. And then here we have the medium-sized EVs, which are showing positivity for phosphatidylserine and coming off at a different pool. This was a method that we developed as an alternative to the differential centrifugation, which you see here on the right. So this is a flowchart summarizing the traditional way, which is the differential centrifugation way of isolating EVs, and the new filtration method that we developed. So starting with plasma or cell culture supernatant, after a low-speed centrifugation to remove the cells and debris. In fact, this is again showing how things have evolved. In those days, everybody was spinning at 25,000 G. We found that you could, at that centrifugation speed, you'll probably be pulling down clumps of exosomes as well. So the smaller exosomes generally stay in the supernatant, but only if you do a gentle sonication beforehand to break up those exosome clumps. So the filtration method simply passes the medium EV SEV mixture through a filter of pore size 0.1 micrometer. The exosomes can be recovered from the filtrate, and the medium EVs can be washed off the filter. So here you can see the medium EVs, which are more PS positive, as seen through an Exin-5 binding, than exosomes. We also show representative EM images showing the expected morphology. And here we see transmission electron microscopy of EVs being released from cells. Now, to be able to call vesicles, microvesicles, and to be able to call them exosomes, you actually have to be very aware of biogenesis and be able to recount it and prove that that's the way they've been produced. So here we see the medium-sized EVs that have been released from this cell, which has been activated. And here in close-up, you can see exosomes, the smaller ones here, and the asterisked medium-sized EVs. So we'll show you a few slides here of this. So here you see the medium-sized EVs and the exosomes. Here again, the arrowheads are showing exosomes. Here you can see this is a bit better in the sense that these were separately purified exosomes. So you can see that cup-shaped morphology. And here you see exosomes and vesicles, medium-sized EVs coming off this cell. And here this is a multivesicular body releasing its cargo into the extracellular space. Here we see a HeLa cell being infected with a metacyclic trypamastigote form. That's the infectious form of the Trypanosoma cruzi parasite. This is a unicellular protozoan parasite. Here you see the kinetoplast. Here you can see the tail. There's vigorous movement of that tail as the parasite tries to get into the cell. And actually, if you see this live, it's very interesting to see the parasite probing the cell, moving from side to side as it's trying to almost look for an area of weakness to gain entry into the cell. Here we see this was a N equals 57 sample of people, and we were looking at the plasma vesicles isolated from them and comparing fresh versus frozen and looking at differences between male and female. This has been often repeated since then. This was looking at the plasma vesicle levels with age, and you can see a slight decrease here, and this is what others have found too. It was slightly surprising at the time, but that is the case. Here, although this was insufficient sample size, there seemed to be a difference between vesicles that we counted in fasting versus non-fasting subjects. So that's probably a pointer as to how these things should be numerated in the future, as with other analytes. This was a bit curious, but again, small sample, looking at non-smokers versus smokers, and we found that smokers actually had decreased levels of plasma EVs. We didn't really follow this up very much other than to look at the effect of nicotine. I know it's only one of the components, but nicotine is anti-apoptotic. So you can see here the white bars are decreasing, showing decreasing percentages of cells in early apoptosis, and the same is found with Calpectin. And as a positive control for something we know will inhibit EV release. So the EV release is being inhibited, and the cells are becoming less early apoptotic. Now, this table is actually looking just at red cell EVs, but the same applies to the general population of EVs that you find in plasma, and that is that there are increased levels with various diseases that you can see here listed on the left. Atherosclerosis, beta thalassemia, renal failure, malaria infection, other infections, meningococcal, and so on. And here, what we see here is for, this was a large study that we conducted looking at Plasmodium falciparum infection in a large number of ill people. And you can see here that the number of vesicles is much increased compared to the uninfected. Now, what's interesting here is that we started to look at this according to age. And you can see that the vesicle levels are raised in the very young and in the old. And of course, that's not that surprising because of the immune status of the elderly and the young. And here we can see that with very small increases of parasite density, there's an immediate increase in the number of vesicles present. And then looking at Chagas disease, these were experiments that we did in mice. Again, you can see increased levels of the vesicles in infected mice, and here over time. And here in an autoimmune disease in lupus, you can see in SLE plasma here on the left, increased levels of vesicles compared to the control. Same theme here, which I'll just rush through. But interestingly, it's only the intracellular pathogens, and even pathogens related. So for example, here in this panel, we see Trypanosoma cruzi causing an increase of extracellular vesicles, as we would expect and showed earlier. But here, the related extracellular trypanosomatid, Trypanosoma brucei, which causes sleeping sickness, does not enter the cells, does not cause a release of vesicles. And the same here with Salmonella typhimurium and a non-invasive mutant, SP11, no release of vesicles. And this is with a virus. So what about microvesicles in autoimmune disease? We saw that the levels of vesicles were increased in SLE. So these microvesicles, or medium-sized EVs, they look like miniature apoptotic cells in terms of the PS exposure. So we decided to look at autoimmune disease in which there may be defective apoptosis of apoptotic cells. Maybe microvesicles play a role. So here, just like an apoptotic cell has the PS exposition, so do the released microvesicles. Could they get in the way of the ability of the macrophage to remove the apoptotic cell? Same sorts of things are going on here with a pathogen here on the left and the host cell it's trying to infect, and the vesicles, could they be being released as some sort of decoy to prevent the pathogen getting to the host cell? Indeed, sometimes these vesicles may actually attach to the pathogen, and that may impart some protection of the pathogen. But going back to the SLE, here we were able to show with the white bars on the right that the amount of phagocytosis of the apoptotic cell is being decreased with increasing amount of vesicles, and here by fluorescence and here by percentage phagocytosis. And when we looked at the antiphospholipid antibody plasma and SLE, we found that there were increased levels of vesicles released in the APLA plasma and in the SLE, as I described before. And here, when we tried to remove the APLA antiphospholipid antibody component from the SLE plasma and tried to use that to stimulate the vesicle release, it was decreased compared to the SLE plasma. And we were able to restore that and so on. So we didn't pursue that work anymore, but it's, I think, quite important, and quite a few people have looked at it and obviously have carried on working in that area. Now, going back to the infectious agent, if I may, and also with this idea of decoy and protection of the pathogen by the vesicles that have been released from the host cell, I want to look for a little bit on Trypanosoma cruzi. And here you've got the infectious form of the parasite. It enters the human host when the triatomine bug takes a blood meal, defecates, the parasite goes in through the feces, it's scratched into the wound. It's not particularly fussy about which cells it goes into, but eventually it does cause damage. And it's the epimastigote form, which is found in the midgut of the bug, and that's the infectious stage then of the human. I think we'll skip that because of time. So what we see here is the microvesicle release over time. This is the kinetics of microvesicle release, invasion, and we've been looking at lysosome exocytosis. So we measured microvesiculation and invasion in panel A, and microvesiculation and lysosome exocytosis in panel B over a 60-minute time period. During the first 30 minutes here, there was an increase in microvesicle release and invasion, but over the next 30 minutes, although there was no increase in microvesicle release, invasion continued to increase up to 60 minutes. During the 30 to 60-minute period, both microvesicle release and lysosome exocytosis remained high but steady. So this is showing the lysosome exocytosis, the bars, and the line graph is showing the microvesicles. And down here you can see that the parasitophorous vacuole, in which the parasite, once it's entered the cell, resides, it's got the T. cruzi in it. Okay, so I think we'll skip this actually, because we're kind of running out of time here. The vesicles released from the host cell attach and indeed perhaps fuse. There's some sort of either fusion or hemifusion, I cannot be sure, but this is an R18 dequenching, octadecyl rhodamine dequenching experiment showing that the metacyclic trypamastigote, that the labeled vesicles are undergoing some sort of fusion or hemifusion, as I said, with the cell. And this is not occurring when the proteins are removed from the parasite, when you're removed with trypsin. So here we can see that the microvesicles that have been released because of the infection, they can actually enhance the infection and even superinfection of HeLa cells by metacyclic trypamastigotes. Here we can see that we're measuring number of parasites per 100 cells and infected cells per 100 cells, which is the black bar and the white bar. And we can see that the vesicles cause an increase in infection. Here they're heat inactivated and they don't. Here there's a dose response. Here there's a superinfection. So we have cells having 2 or 3 parasites in a cell. And that's only happening when with the microvesicles. And of course here we're seeing this increased infection in the presence of vesicles for the metacyclic, the infectious form, but not the epimastigote, which is the form found in the insect. And here we see that the type of vesicle that you add is important, and it's important in the sense of how much TGF-beta it's got. I won't go into that, but it's known that TGF-beta enhances the infection of Trypanosoma cruzi. So here you can see different cell lines, and they have different amounts of TGF-beta. And in fact, the T cell line vesicles seem to have the most enhancement of infection here because it has the highest level. Here we were able to block that with an anti-TGF antibody and block, in terms of SB431542, the TGF-beta signaling, and then the infection goes down. And we can skip straight to the animal model. Oh, this is all done in mice, actually. There's all these experiments. And I just wanted to show this is a typical curve. It has this unusual shape of the infection. And so we see here the number of parasites, and then it falls. And this is, I can't quite see what I've written here, but that's with vesicles, yes. So that's with vesicles. With vesicles, you've got this enhanced infection, and without vesicles, it's much lower here, the lower graph. And the other thing that these vesicles are doing, it seems, are protecting the parasite from complement. So here you can see in this graph, we've added the vesicles, and the lysis, I think that's lysis inhibition, it's increased. Not with the heat inactivated vesicles. And here you can see that when you add these vesicles, that the amount of complement that's deposited on the parasite is reduced with increasing vesicles. So here we're measuring C3b deposition. So what's going on here is the parasite is stimulating vesicle release. There's some sort of damage to this plasma membrane. The parasite gains entry, and eventually it forms this parasitophorous vacuole where it undergoes acellular replication and so on. Just a curiosity here, we also looked at the microvesicles. Again, host ones seem to have no effect on this parasite, Giardia intestinalis, which is a gut parasite. But the parasite itself makes extracellular vesicles, as we can see here, in the presence of bile. And these are vesicles which are annexin 5 positive. And interestingly, what we also found was that these parasite vesicles aid the attachment of the parasite to intestinal epithelial cells, as we can see here with increasing amounts of the vesicles added. In fact, having done this experiment, I thought it would be interesting to try this out with a bacterium, which also attaches to the same sort of intestinal epithelial cells, the Helicobacter pylori, but we were beaten to it. That does indeed aid attachment as well. So just summarizing this paper, we showed that Giardia intestinalis medium-sized EVs increase the parasite adherence to the intestinal epithelial cells and uptake by immature dendritic cells, and that may activate the immune response against these parasites. So finally, I'd like to thank the people that worked with me in the early days. Many of these are PhD students, and because this work was done during this time, I've listed the people that have helped throughout in this work. And I think I better stop now, otherwise there'll be no time for questions. Thank you for your attention.
Thank you very much, Jamil. Beautifully presented. And our first question is already there before I finished. It's Hamid. Hamid, the floor is yours. Turn your microphone on. Thank you very much.
Okay, thanks very much, Jamil, for this very, very interesting talk. And it's really, really very good to see you back at London Met.
It's lovely to be back.
Thank you. Your introduction to microvesicle, you said was in 1999, so more than 20 years ago. And my introduction to microvesicle, although since the day I joined London Med, I always associated microvesicles with you and your work, but I never really had a chance to have a look at it until 20 days ago when I was asked to read two RD2 reports from our PhD students, and I initially said, okay, this is not my area, but as I started reading that, I got very interested into that, specifically because most of the attention until recently has been on the gram-negative bacteria.
Yes,
but it seems that it's picking up for the gram-positive bacteria as well. One of my main question is that knowing that the gram-positive bacteria have got a thicker peptidoglycan layer than compared to the gram-negative ones. Does it not make it very difficult to isolate the microvesicles first? That's my first question. And secondly, I really see a potential in the work that you do and what I do with probiotics. And my interest comes from the fact that probiotics are mostly gram-positive bacteria. I know some people suggesting some gram-negative bacteria, even E. coli, could be used as probiotics. I'm very kind of cautious not to go into that area because they are gram-negative. But with the gram-positive bacteria, I think if that's the case, if we prove that that is the case, that microvesicles have got a role in that, and we are able to isolate them, it would be interesting to look at the probiotics from this point of view and how they could interact with other organisms as well as other cells. So organisms like potential pathogenic microorganisms and cells such as epithelial cells. If you could elaborate a bit more on this.
Yes, it's an absolutely interesting topic, Hamid. Thank you for these excellent questions. And I'd like to start by saying that in a lab meeting over 10 years ago, well, 13 years ago, somebody asked whether they thought that bacteria could make vesicles. And I remember myself saying, oh, come on. That's not likely. You know, they've got, as you said, they've got this thick peptidoglycan wall and so on. I thought, no, but actually they do. And you're right to say that much of the work is being done on gram-negative. And the reason for that simply is that it's easier to isolate these vesicles, I think. And there's a plethora of work on that, but very, very little on the gram-positive isolation. But there are lots of different ways that these vesicles are released from bacteria. And of late, there was a very interesting article saying that there's an explosive release of vesicles, which actually, they're not released as vesicles as such. They recircularize into vesicles, but there's this explosive formation. And yes, I think it would be, in fact, I mean, you need to use slightly different techniques to that. You can get a crude isolate of vesicles by using differential centrifugation, but there's ultrafiltration and other techniques to isolate this. In fact, that project looking at the microfluidics is using, it's being used to isolate bacterial vesicles. So those are the samples that we are providing for the bioengineers to develop the microfluidic system are bacterial vesicles, in fact. So yes, this is a marvelous area. And yeah, Clostridium difficile makes EVs, bacterial EVs, and they're important, as you hinted, on their interactions with host cells, because they can pass through the intestinal barrier and move into the circulation, and can actually even pass the blood-brain barrier and have effects there. So in the brain. So yes, bacterial vesicles are incredibly interesting, and I have an ongoing project, 2 projects ongoing in that area, and I would love to do more. So let's work together.
Thank you, thank you both.
Sure. I'm up for it. Thank you.
Great.
It sounds like we've already got it's a speed dating session is underway. Our next question comes from Kevin Devine. Kevin, the floor is yours. Turn your mic on.
Yep. Okay. Can you hear me, Jamil?
Yes, Kevin. Loud and clear.
Yeah. Okay. Yeah. Thank you for a fascinating talk, a really good overview of your work with microvesicles. My question concerns virus infections, like, for example, HIV. Do you see elevated levels of microvesicles in that infection? And if so, are the microvesicles, how similar are they in terms of protein shape and amino acid sequence to the glycoprotein on the viral coat? And are they then good decoys for to prevent sort of hunter-killer cell attack on infected CD4 cells? And the same thing could apply to cancer cells too. You know, emission of microvesicles to sort of trick the immune system.
Yeah. No, I think the vesicles from infected cells will carry some kind of viral signature. So that is most likely the case. And the first point, whether there'll be increased levels, almost certainly. We've shown that in vitro. I believe in vivo too, these have been recorded. I was actually curious for whether anyone's looked at this with the SARS-CoV-2 actually, but I've not read anything on that at all. And yeah, the whole idea of decoys is very interesting, and I think that's one of the tactics that intracellular pathogens use to ward off, also to ward off the immune effector mechanisms and so on. It's a two-way sort of interaction actually of pathogen vesicles and host cell vesicles. Now, of course, the viruses don't themselves release vesicles.
Yeah, that's right.
And I mean, what's interesting, and I know Gary's listening, and that we found many years ago that the vesicles released from infected cells infected with non-enveloped viruses, that the viral capsid could actually, or components of it, or actually the RNA even, could be found in the vesicles.
Wow.
And so that's another area that's of interest actually.
And then if I could ask one quick question, another one about cancer metastasis. I presume if you've got nucleic acid sequences in vesicles as well, is this a primary mechanism of metastasis in certain types of cancer cells? Emission of microvesicles? Yes.
So what we've looked at in terms of microvesicles released from primary tumor cells is one thing that, I mean, firstly, I would like to say one of the most seminal studies in this area has been looking at the vesicular release from primary tumor cells and how they're involved in setting up a pre-metastatic niche. And that also another interesting topic in the vesicle world is to do with targeting. And this study on cancer metastasis actually began to give us some clues as to how vesicles in general may target other cells. And it was down to the particular array of integrin molecules that were expressed on the exosomes as to whether they would go from prostate to bone or wherever. So you could actually delineate where they would go according to the particular integrins they expressed. So there, the exosomes are setting up a pre-metastatic niche. Also, exosomes are involved, vesicles, I mean, are involved in epithelial mesenchymal transition, and that's to do with TGF-beta that they carry, which is activated and stimulates this process. And this is, of course, loss of the adhesion proteins on these primary tumor cells and the beginning of the metastasis. Does that answer your question a little bit?
I think yes, indeed. Thanks very much indeed.
Yeah, well,
thank you. Great talk.
Thank you both. Our next question is from Li Jun. We have about 7 minutes left, so jump in if you wish to ask a question. Li Jun, the floor is yours.
Hello. Yes, it's a very nice talk. Yeah, indeed.
Thank you. Nice to hear you.
Can you hear me well? Yeah.
Yes, thank you.
Yeah, this certainly this is a very hot areas of research and have a lot of the clinical and therapeutical applications. And I didn't know this too much, but I just wonder if you could give me some, slightly update or something about the structural function of the vesicle itself, or is any research on this side, or what's the difficulty part in doing this? Yeah, thank you.
Yeah. So I think this is a very interesting question, actually, Li Jun, because I found that, and this is really symptomatic of the way research is funded, is that we are always pushing ahead to try and find applications of our work. We're being asked in our grant applications, how can this work be made translational within 5 years? And as a result of that, push in that direction, in this field in particular, which has been so incredibly fast moving, and even more so after the International Society of Extracellular Vesicles was founded in 2012, is that much of the fundamental biology has been left behind.
Yeah.
And then we are playing catch up because we need to understand it to be able to make the work translational and to derive therapies. So, for example, with the targeting, that was an issue. Now, of course, we can genetically engineer vesicles to express a ligand which will target them to a particular receptor. So, for example, the first case of that was in the early 2010s, where mouse dendritic cell exosomes were isolated and genetically engineered to express a ligand which will target them to the brain. And they were carrying silent RNA to knock out base 1, which is involved in Alzheimer's disease. So the basics of how they are being made, the pathways of biogenesis are not fully derived. The way of characterizing these vesicles is continually being debated, and the parameters are being changed slightly all the time, as you can see just with the terminology.
Yeah.
So the basic fundamental biology is needed to be understood. How we package, whether it be silent RNA or a chemotherapeutic agent, into the vesicle. Do you put it into the cell and then it gets taken up into the vesicle? Do you put it directly into the vesicle? For example, the silent RNA knocking out the base 1 was electroporated into the vesicle, but subsequently it was found that they were not actually inside the vesicles at all. They were tethered to the outside, but they still carried out their function.
Yeah.
So you know, I am very much, I've always got one eye on the basic biology of the vesicles. How do they interact with their cells? Are they taken up by endocytosis? Do they attach? Are cells on the surface of the vesicles activated so that they can then interact with cognate receptors on the recipient cell? Do they get taken up by macropinocytosis, or a combination of several of these with any one particular cell? So all of this is work that we are sort of catching up on, because of the drive to make everything therapeutic.
Yeah. You know, even for the kinetic of the release itself, it's quite a lot of things can be done, I think. Yeah. Excellent.
Absolutely.
Yeah. Thank you.
Thanks, Legion.
Thank you, Legion. Thank you, Jamila. And a question now from Rena.
Thank you. Hi, Jamila. Thank you so much for this amazing talk. Can you hear me?
Thank you. Yes, absolutely.
Okay, right, good. So just a couple of questions that you may have mentioned that in your talk already, and I might have missed it. So you said in your presentation that EVs are released after 20 or 30 minutes after stimulus of 20 to 30 minutes. How long after the EVs are stable for?
So Rena, they start being released. In fact, some other work showed that even after 2 minutes, they started to already be released. It's that the rate of increase of release is itself, you know, there's increasing amounts being released over that period, and then there is no further increase of release. And in fact, cells can, if the stimulus was taken away, recover and go back to a resting state. So you are asking about the vesicles once they're released. In effect, are you asking about half-life, or I couldn't quite understand?
Yes, half-life.
Okay, that's a question that was asked. I remember people going around asking that in 2012, and I still don't really know the answer. I mean, what I know is that vesicles, you know, tend to end up in the liver, and I guess a lot of therapeutic ones will probably end up there initially as well. So what I would also say is that compared to the other kind of vesicle, if you like, that could be used as a drug delivery vehicle, for example, liposomes, I would suggest that extracellular vesicles, whether they're small or medium, have a much longer half-life. I mean, they're much better suited to being protected from things like complement attack and so on, because they carry the complement inhibitory proteins and so they are protected. So extracellular vesicles, the absolute answer to that, I do not know. What is the half-life of these vesicles? I do not know. All I know is that we've been able to do experiments where we've added vesicles to a mouse, and you know they've achieved the effect of increasing parasitemia or what have you. There didn't seem to be any untoward effects on the vesicles in vivo, but the actual answer to that, what is the half-life, I cannot categorically say, and it's partly because I just haven't been reading about that. But I know that has been, and probably somebody has looked at it and has got an answer somewhere. but I don't know what it is.
But I guess your answer to that, they have been administered to mice, and they saw, you know, a proper response. That
Yes.
I think that will do for me. Thank you.
Yeah. Okay.
And just a last question. Sorry, Don.
Yes.
That's all right. This is then the last question. No problem, Rena. Carry on.
Okay. So you also mentioned that the AV release can be inhibited by calpectin. Okay.
Yes.
So what happens if we have AV inhibition of the release? So what happens to the host cell? What happens? Does the cell viability change, for example?
Yeah, of the cell once it's, yes. I'm trying to remember what we saw with that. Maybe I could ask one of the other guests who I know might know the answer better to that.
Yeah. Is there any effect if we inhibit EV release?
Yeah, I know. It's a good. I know what you mean, and well, that points to the other thing, doesn't it? Which is the other idea of vesiculation in general. Why cells vesiculate? It's to probably get rid of things like misfolded proteins that they don't need. So it's a kind of maintenance release of vesicles to maintain the normal cell physiology. And in that case, I can see what you're saying, that if you inhibit vesicle release, it might be detrimental to the cell. That's probably quite an interesting thing to look into if someone hasn't already done it. I mean, I must say that almost every idea that one has nowadays, if you look into it before you start, which obviously you should, you'll find that it's probably already been done. But that's a good question as well. And I again, exactly. So Samira was the one person I was going to ask actually for help with this. She said about getting rid of waste. Exactly. I mean, that's one of the first ideas for why cells release vesicles. It's to do that.
Yes. Do I have time? Hi there.
Yeah, of course you do. Yeah, and then we'll definitely wrap after this. Carry on.
Okay. So Rena, the answer to your question is we've done some experiments when we inhibited release of microvesicles using calpectin, and we could see clearly, for example, when we treated the cells with apoptotic agents, chemicals that induce apoptosis, we could see that when we inhibit release of microvesicles, we could see higher percentage of cell death, because this is one way of removing the like damaging agent by releasing the microvesicles, or basically apoptotic signals that contribute to the whole process. So if you treat them for cell death and you inhibit release of microvesicles, you see increase in microvesicles. If you look at the control cells comparing to the cells that were treated with these chemicals, you can clearly see that the viability of cells is higher when we don't inhibit release of microvesicles. If it makes sense.
Yeah.
Yes, no, it does make sense. Yes.
It depends what the stimulus is for the vesiculation in the first place. So if it is like Samira said, if it's something that's inducing apoptosis, then obviously the cell will try to get rid of it. And if you inhibit that, then it's not good for the cell. If it's probably something like sublytic complement, which forms pores in the plasma membrane, but not enough to induce lysis, and that this is something that's recoverable indeed by vesiculation itself, because you can release the deposited MAC, membrane attack complex, then that is recoverable. And as we saw, that everything went back to a sort of normal state. So I think it depends. If you inhibit vesiculation, it depends what the stimulus for the vesiculation was in the first place.
Thank you very much. Thank you, Jamil. Thank you, Samira.
Thank you, Samira. Thank you, Rena.
You're welcome. No problem.
Thanks.
Thank you, Jamil. Thank you, Samira, Rena, and all the other questioners and contributors. That was really excellent and stimulating talk. Thank you. Great to see research has been going on so long, so deeply, and in so many areas, and yet still has so much, you know, so many answers to give and so much more research to do. So clearly, it's going to keep you going for a lifetime and a bit more, which is probably the noblest sort of science when you lay yourself out like that. But this was very, very interesting. Just to say that our next talk is in 2 weeks, and we have somebody else from biosciences, Gary McLean, who's going to talk to us about viruses and the hunt for vaccines. Mavis put up the link there in the chat, so you can go and book your ticket now. Thank you so much for this, and we're looking forward to hearing another of your comrades in a couple of weeks' time. So in the meantime, everyone, stay safe and remain vigilant. We may be opening up, but it's still out there. Good luck, everyone.
Thank you, Don. I'm trying to send a chat message, but I don't think it's getting through. And I wanted to say thanks, everyone, and that it's lovely to be back.
It's great to have you back, and that's very clear from the questions and some of the comments. So welcome back, and you know, much more research like this down the track. Good luck.
Thanks, Don, for organising this. As Legion says,
no
it's
reason. Oh, actually, thank you to particularly to Maeva, who's really the engine room of organisation behind this.
Yes,
see you
thanks,
all, everyone.
Maeva.
Take care.
Bye, everyone.
Bye, everyone.
Bye.
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 Bye-bye.
Bye.
Some lovely comments in the
Bye,
chat
Benny,
there.
Yeah.